Biochemistry is considered as one of the most important areas of research that yield far-reaching discoveries, combining the core concepts of various disciplines, primarily chemistry and biology. Biochemistry plays an essential role in the development of groundbreaking scientific methods and approaches. Biochemical research is directly linked with the development of drugs, therapies, and methodologies that might cure the diseases that are not remediable nowadays. So, I think watching this video will give you insights of how biochemistry works in the fishery industry. So, a pleasant day to everyone. My name is Willie May Ann Asepi. So, today, we will give you 10 published research that me and my co-researchers have picked. So, please keep on watching.
The title of the research is Influence of Different Extraction Methods on Fatty Acid Composition of Lipid Extracts of Chlorella vulgaris Behring from Laguna de Bay, Philippines. The authors are Rochelle Joey Isaracanlao, John Owen Nacorda, and Hedelisa P. Hernandez. And it was published in March 2019. And that was conducted in Agricultural System Institute, College of Agriculture and Food Science, University of the Philippines, Los Baños College in Laguna, Institute of Biological Sciences, College of Arts and Sciences, and the University of the Philippines, Los Baños, Laguna, Institute of Chemistry, College of Arts and Sciences, and the University of the Philippines, College in Laguna. The instruments are sampling and statistical, and the DOI number is shown in the screen. Analysis on the strong and weak parts of the research. In the strong parts, the researchers use different extraction procedure on the fatty acid to further analyze the composition of lipid extracts of Chlorella vulgaris Behring. The researchers construct a flow diagram in order to optimize the extraction of fatty acid from Laguna de Bay. The researchers construct a pie graph to view the profiles of the fatty acid composition of C. vulgaris lipid extracts analyzed by GC with flame ionization detection is widely used for lipids analysis. Overall, the results show that different extraction methods lead not only to different lipid yields but also influence the fatty acid profile to a great degree. On the weak parts, though there are numerous reports regarding the lipid extraction in Chlorella and its fatty acid composition, there have been no literature on these parameters for the C. vulgaris from Laguna de Bay. There are also no local studies conducted relating the different extraction methods on the corresponding fatty acid profile. Thus, the objectives of this study were to optimize lipid extraction from C. vulgaris and characterize the fatty acid from the different solvent systems and extraction procedures.
Local published research is entitled Fatty Acids in 6 small pelagic fish species and their crustacean prey from the Mindanao Sea, southern Philippines. So the authors of this study are Efrain Bicoy Mitilio and Ana Arlene Aspiras Eya. This study was published on August year 2014. So this study is conducted at Mindanao State University in Liga. both fatty acids has been attributed to their use as a major source of energy for metabolism and growth. In addition, this study demonstrates that fatty acids are useful trophic biomarkers. However, other techniques, including stable isotope analysis and DNA-based techniques, may also be helpful as alternative approaches in elucidating trophic linkage among species analyzed in the study. However, the weak part of this study is that species from warm waters tend to show high levels of palmitic and stearic acids compared to those cold waters. These differences is due to metabolic differences between cold and warm water species, because these fatty acids are not usually subject to differences in diet.
Chemical composition and antioxidant properties of Philippine Chrysostria eridale residue is one of the local published research that can be found in the internet. The authors are Ernestina M. Peralta, Carmela Jean Imonaya, Rhoda Mrs. Mora, and Augusto E. Serrano Jr. It was published on July 3, 2018, and the study conducted at the Institute of Fish Processing Technology, College of Fisheries and Ocean Sciences, University of the Philippines, Visayas, Maigo, and Iloilo. The instrument used in the research is experimental. Now let's go to the analysis of the strong and weak part of the research. So, for the strong part, the study discovered that oyster residue can be a potential source of natural antioxidant that can be used to replace synthetic antioxidants, thereby preventing free radical-induced lipids preoxidation, which is responsible for the formation of flavors and undesirable chemical compounds in food. As a byproduct of oyster extract processing, oyster residue was discovered to have a high protein content, essential amino acids that is required for the human body, and bioactive compounds with antioxidant properties. For the weak part, the low moisture content and water activity of the product can inhibit growth of most bacteria, molds, or yeast, thus suggesting its shelf life stability.
Good day everyone, and my report in local research is about the lactic acid bacteria in Philippine traditional fermented food, and the authors are Charina Gracia B. Banaay, Marilyn P. Balolong, and Francisco B. Iligado, and it is published on June 30, 2013. And the studies was conducted in Central Luzon, which they have fermented rice and fish mixture, and there was also fermented pork and sweet potato that involved in this study, but located in Eastern Visayan region like in Samar and Leyte. And the instrument used in this study is the process of fermented products through quantitative analysis. And the strong parts of this study was the many benefits have been ascribed to probiotics. For example, Lactobacillus testing have been shown an effective in preventing diarrhea due to enterotoxigenic Escherichia coli and cholerogenic Babesia using rat. This may be accounted for by its ability to kill the pathogens and inhibit further growth, because the probiotic is used in 2 studies mentioned in a commercial strain. But current research on probiotics is progressed to the research for indigenous lab for use in the development of locally produced functional food and investigation of their utility for biomedical application. And the metagenomic approach to investigating lab present in fermented food have been shown the diversity of potentially beneficial species present other than the already detected by the conventional culture-based method, and the development of the functional food production and a potential in disease management. And the weak part of this study was the antimicrobial properties have been investigated in several studies, but although pediosins and plantarisins promise that their application are limited, at present because it is well known at the fact that other bacteriocins aside from the nascents are not yet approved for food use. But for pediocins and plantaricins, the most practical use for now would be in dermatological and animal healthcare use.
Local published research titled Polyunsaturated fatty acid contents of some traditional fish and shrimp-based condiments of the Philippines. The author of this study, Nemesio Montaño, Grace Gavino, and Victor C. Gavino, published on January 11, 2010. The sample study were taken in Marine Science Institute, University of the Philippines, Quezon City, Philippines. The instrument that used is fermentation. Next is the analysis of the strong and weak parts of the study. Strong. The research used different procedure of ash and fat contents of fish and fish-based condiments. To prepare the condiments, they are incubating the fish, shrimp with high concentration of fat, salt, and high humidity at ambient temperature over several months. Weak part. This particular condiment were not significantly different from the fresh, untreated shrimp fry, indicating the presence of mechanisms that protect against polyunsaturated fatty acid oxidation during the preparation process. Also, the DHA contents of condiments derived from juvenile Siganos approach that of the acetes condiments because of its relatively high fat condiment.
Let's proceed with 5 international published research. So the first international published research is entitled Fatty Acid Composition and Lipid Content in the Copad Limnocalanus macaroons during summer in the southern Bosnian Sea. So the authors of this study are K. Makinen, M. Elfbing, J. Haninen, L. Laaksonen, M. Rahasilta, I. Borinen, and J.T. Somela. This study was published on the 26th month of June in the year 2017, and it is conducted at La Rollenfine Chemicals AD Malmö, Sweden. So the instrument that is used in this study was hydrological and plaston sampling. Also the DOI number, again, can be seen in the lower part of the screen. Okay? So now, I'm going to share with you guys the strong and weak part of this study. Okay, so let's start with the strong part. So the strong part in the study is that in the Bosnian Sea, Limnocalanus macaroons has been suggested to be a key species responsible for improving the physiological condition of herring in spring and early summer. As shown in this study, the wax ester content of an adult Limnocalanus macaroon is higher than that of Pseudocalanus acrospes, the preferred prey species of this herring in the central Baltic Sea. In spite of the apparent large energy reserve, this study concludes that the summertime wax ester content and survival of adult Limnocalanus macaroons seems to be connected to the abundant availability of good quality food. This is important in the face of the ongoing climate change, as climate change scenarios for the Baltic Sea predict that the plankton community will change towards smaller size and poor quality taxa as a consequence of a further decline in seawater salinity and an increase of water temperature, stratification, river flow, and nutrients. However, the weak part of this study is that this study does not tell about the interannual variation of the wax ester content in Limnocalanus macaroons, and no such information is found, to our knowledge, in the literature either. Therefore, interannual data on the wax ester content and fatty acid composition composition of Limnocalinus macaroon should be collected in order to foresee these changes and to provide a more accurate picture of the tropodynamics.
The title of the research is Lipids in Marine Ecosystem. The author is Christopher C. Parrish, and it was published in February 13, 2013. And the study was conducted in Memorial University of Newfoundland, St. John's, Canada. Sampling and optimization are the instruments that he used in the study. And the DOI number is shown in the screen. So let's proceed to the analysis on the strong and weak parts of the research. In the strong part, the researcher used to determine production of biogenic material of dietary value to marine organisms as well as to indicate water quality. While the emphasis here will be on biochemical and ecological aspects of marine lipids, the most stable compounds have also been extensively used in oceanographic studies. Lipid marker determination in sediment cores can show the sensitivity of sediments to change land use patterns near the land margins. In the weak parts, lipids provide the densest form of energy in marine ecosystems. They are also a solvent and absorption carrier for organic contaminants and thus can be drivers of pollutant bioaccumulation. Among the lipids, certain essential fatty acids and esterols are considered to be important determinant of ecosystem health and stability. Fatty acids and esterols are also susceptible to oxidative damage leading to cytotoxicity and a decrease in membrane fluidity. Marine lipids are also a valuable tool to measure inputs, cycling, and loss of materials. Their heterogeneous nature makes them versatile biomarkers that are widely used in marine trophic studies, often with the help of multivariate statistics to delineate carbon cycling and transfer of materials.
Polyunsaturated fatty acids in fish tissues more closely resemble algal than terrestrial diet sources is one of the international published research that can be found in the internet. The authors are Naden M., Fen Go, Michael T. Brett, Stuart Bond, and Martin J. Kites. It was published on November 16, 2020. The study conducted at Functional and Evolutionary Ecology, Faculty of Life Sciences, University of Vienna, Australia. The instrument used in the study is observation, experimental, and sampling. The DOI number is attached in the screen. And now, let's go to the analysis of the strong and weak part of the research. For the strong part, researchers has highlighted the nutritional importance of high-quality algae. Algae provide physiological important omega-3 polyunsaturated fatty acid, particularly eicosapentaenoic acid to consumers. Furthermore, this field study demonstrates the nutritional importance of polyunsaturated fatty acid for neutral organs and aquatic consumers of headwaters. For the weak part, most terrestrial plants and benthic streams algae are deficient in the long chain. Entry polyunsaturated fatty acid
International research was the title Latitudinal Change in Lipid Content and Fatty Acid Profile of Juvenile Female Red Spot Lobsters in breeding areas of the Humboldt Current System, and the authors are Fabian Guzman Rivas, Marco Quispe Macaca, Dante Quirello, Mauricio Ahumada, and Angel Orzua, and it is published on June 22, 2021. And this studies was conducted in Chilean coast. This is along the western coast of the southern cone of South America, and Coquimbo coast. This is bordering Argentina to the east and fronting the Pacific Ocean to the west. And in Concepcion coast was also located in Madrid or South Central in Chile. And this is near in the mouth of the Vibio River. And the instrument used in this study was quantitative biosampling and statistical. Let's say, for example, in quantification of lipid content, for extraction of lipids, it's about, let's say, 20 milligrams of the dry weight of viscera and 20 milligrams of muscle were used. And the total lipid analysis was performed using the standard method. And the standard method uses dichloromethane and methanol as an organic solvent for lipid extraction, with potassium chloride as an ultra-pure water solution. And the fatty acid profile, after analyzing the total lipid content, the fatty acid profile, like for example the acid methyl or the esters work was qualified. And then this was done by the following the standard method with slight modification, which uses inhexane as an organic solvent of fatty acids. And the strong parts of this studies was because it increased the knowledge regarding the spatial variation of these bioenergetic reserves like lipids and fatty acids, and in addition to other biomedical parameters like for example in glycogen, proteins, amino acid, digestive enzymes, and of juvenile population could help predict the bioenergetic condition and nutritional quality of the future exploitable population. Within an ecosystem approach, this data could also aid in assigning catches to certain population, for example in fisheries traceability, in developing strategies of exploitation and sustainable management, especially in areas where the resource has larger size and better nutritional quality. And the weak part of these studies was they did not find such positive directly or proportional relationship between body size and latitudinal, which in turn latitudinal or latitude, which is supposed to be their title.
International published research titled Comparative Study of the Chemical Composition, Fatty Acid Profile, and Nutritional Quality of the Piocoelurus alexandrae, a Brazilian carnivorous freshwater fish grown in lotte, lentic, and aquaculture environment. The author of the study, Lee S. Sante, Anal, Adrian A. Iwamuntul, Daniel V. Cripapalde, Tulio Boaventura, Lilian V. Tizera, Edgar A. Tizera, and Ronald K. Luz, published on December 2, 2019. The sample study were taken Sao Francisco River, located in Minas Gerais State, Brazil. The instrument that used sampling. Next is the analysis of the strong and weak part of the study. Strong part. According to the research, cultured and lentic fish had significantly higher lipid levels than lutic fish. Lentic fish had greater portion of a fatty acid and lutic or a cultured fish alexandrae through restocking and the development of commercial projects of aquaculture. Weak part. The researchers elaborated the study of evaluate how climate change in Brazil and how it affects the lipid metabolism of Pacamia in different environment. Species is a lentic and lutic environment after water stress, as well as the effects of prolonged thaw in nutritional.
After studying and reading this research, I've learned on what really the importance of and purpose of the research. Since our locality is facing crisis because of the wrong things people do, I would like to take an advance to make a research to solve the problem. Here in our locality of Tagitik Kapatagan, Lano del Norte. As a fisheries student, I know better the importance of marine life, and that is why I decided to make a research to solve the problem entitled Illegal Catching of Small Crabs and Small Fish here in the locality of Tagitik Kapatagan, Lano del Norte. Due catching small crabs and fish, there is no crabs and fish continue to grow and reproduce. What's happening is that the crabs and fish are gradually being depleted. And this is my future plan in my research. And I'm interested to solve this kind of problem.
And first of all, I'm very thankful for the previous studies or research that I've topic in the international, which the title Latitudinal Change in Lipid Contents and Fatty Acid Profile of Juvenile Female Red Scots or the lobster. Because of that, I came up to the ideas that what if I do research about the most popular crustacean in the Philippines, particularly in crabs and shrimp. I will do any studies about that, or I will going to identify how much the lipid content of that particular crustacean in according to their size and what month they catch. And if I've succeeded to that studies is I will going to post that in every restaurants, especially to the restaurants that caters seafoods or mga crustaceans, so that the people or the consumer or the customers to that restaurants is aware how much the lipid contents of that particular food. And I know that was very important for especially to those peoples that had maintaining their diet.
Hi, I would like to share to you guys my future research plan. My future research plan would be tackling about developing, improving, and management of inland fisheries resources in the Philippines. I think it is the best way for us to prevent the threatening loss of our aquatic species. Most places here in the Philippines are now experiencing this kind of crisis, particularly here in my place. Most people who reside in here are fishing is the primary source of food and income. That is why, over time, really felt that marine resources are now gradually disappearing. Through this research, I can able to find solutions on that particular problem so that we can prevent this threatening loss of our aquatic species and preserve them to the upcoming generations.